Today we will be covering transcription. Does anybody know what transcription is already? Go one sentence. Yes, so that's what it is exactly. It is when you use the information stored in DNA to create RNA. Right. We are going to start with some experiments. So, we're going to start with Garrett's hypothesis and the Beadle and Tatum experiment. Alright, so in the 1900s, what did they think of inheritance? So, in the 1900s, the understanding was that only physical traits could be inherited. So, for example, your hair color, your height, stuff like that. But we didn't understand how genes related to physical traits themselves. So, we knew that, I think by the late 1900s, we knew that, well, DNA was a genetic material. And we knew that people had specific phenotypes, but we didn't really have a connection. Uh, Garrett, in 1909, looks like, he studied patients with an inherited condition called alkaptonuria. Alkaptonuria. Not too sure how to pronounce that, but basically it is when your urine is black due to the buildup of something called homogentisate or alkapton. And that darkens when exposed to air, so it makes your urine very dark. What he hypothesized or postulated was that the urine was black due to an inability to break down alkapton, and that breakdown is normally catalyzed by an enzyme, but in people with alkaptonuria, that enzyme is not working. And he postulated that inheritance is because the parent passes on a faulty gene that is supposed to produce the enzyme. So if the parent passes down this faulty gene, then all the offspring downwards will also have this faulty gene, which is why alkaptonuria is inherited. So summarized, it is that the disease is because of an inability to make an enzyme, and he called it inborn errors of metabolism. Garrett was the first person to suggest that inherited physical traits are because of genes that code for enzymes that then catalyze specific chemical reactions. So he linked inheritance or genes to specific chemical reactions in cells or specific metabolic pathways. Um, oh, is someone echoing? Oh, thank you. So his hypothesis was that genes would create enzymes, which would create phenotypes. So this was his hypothesis for the link between genotype and phenotype. Pretty good hypothesis. Yeah. So we know now that Garrett was actually correct. Alkaptonuria is due to a malfunction in the tyrosine catabolic pathway. So in the 1930s came Beadle and Tatum. So they won a Nobel Prize for this specific experiment. It's very insightful. So they performed research based on Garrett's hypothesis and Garrett's ideas. They proposed that genes were responsible for producing enzymes, and they wanted to expand on the one gene, one enzyme hypothesis. Or I think Garrett proposed that genes were responsible for producing enzymes, and he had the one gene, one enzyme hypothesis, but they took it further, and we'll see how. They actually, I think this is one, this is the experiment, sorry. They experimented using the bread mold Neurospora crassa. It's a very simple organism, has a short life cycle, and only requires access to a few biological substances to live and grow. And it could therefore grow on something called minimal media. Since this mold could grow on so few substances, they hypothesized that It has to be able to synthesize all other vitamins and essential nutrients on its own. So in order to do that, it also must have the enzymes to convert these simple substances into essential nutrients. So these were the simple substances: salts, sugar, and the vitamin biotin. So what did they do to test this hypothesis? What they did was, first of all, they mutated the genes in the mold. Then they looked for mutations that affected the mold's ability to make the essential nutrients, and then they took it one step further from there. So what did they do? They used X-rays to basically mutate the genes, create mutants. They used a specific selection process to isolate mutants, and then they created a process to identify mutants based on what nutrients they could or they needed. How do they make mutants? They just treated mold with X-rays to mutate the DNA. In 1927, Hermann Muller showed that X-rays cause mutations in genes. This must have been a lot of work, a lot of work, a lot of trial and error. Step 2, selection. They looked for mutations, specific mutations, where mold could grow in complete media, but could no longer grow in minimal media. Right. So after they have the mutants that could no longer grow in minimal media, but could grow in complete media, what they did was they found, okay, so I mean, same thing as the previous slide. Yeah. Okay, so let's say that they have this mutated mold that can grow on complete media but can't grow in minimal media. What does this mean? Any ideas? Yeah, that's exactly what that means. That's what they thought. So, what should they do next? Any ideas? That's fine. So, what should they do next? This is what they thought about doing next. Exactly, Nathan. Try to figure out what can't be converted. So, this is where supplementation comes in. The specific mutant that could no longer grow in minimal media, they took it and they figured out what nutrient was required. So, how did they do that? First of all, they create a control group, negative control. Then they add minimal media plus vitamins, minimal media plus amino acids, minimal media plus nucleotides, and then they do a positive control. So what does this mean? If the mutant grew with plus vitamins, plus amino acids, or plus nucleotides, then that means that the mold lacked the ability to make that nutrient. If that makes sense. So that one pathway that was previously destroyed, once they supplement it and it grows, they know that that one supplemented nutrient was what the mold could no longer make. So let's keep going. Complete the worksheet. Oh, let's see. So you can't really answer this side, so we're gonna move to this side here. So what did they find? This is what they found. They found that this mutated mold that couldn't grow in minimal media, but could grow in complete media, they found that this mold could grow in minimal media plus amino acids. So what does that mean? That means that this mold, whose genes are mutated, there's a pathway that worked before but could no longer work that created a specific amino acid. So what do? They figure out which amino acid it is. A lot of work. They test all of the amino acids, and finally they realized that this mold was missing the ability to create arginine. Minimal media is over here. Minimal media is salts, sugar, and the vitamin biotin. So in normal mold, they have all the enzymes or they have access to all the metabolic pathways to create essential nutrients on their own with just the minimal media. Complete media would be here, let's take a look at here. Complete media is salt, sugar, and all vitamins and amino acids. So the difference between the two of them is for minimal media, in order to survive and grow, the organism has to be able to convert salt, sugar, and the vitamin biotin into all the other amino acids and vitamins. So going back to this slide, this mutated mold has lost the ability to create arginine. So what did they conclude from all of this? They concluded that mutants that didn't grow in minimal media, but grew with the specific nutrient, which in this case is arginine, meant that the pathway responsible for making that nutrient was broken. So this mold, the pathway to create arginine was broken. Thus, the malfunctioning enzyme was due to mutations in genes because they mutated genes to create this mold in the first place. And thus, genes are responsible for enzyme production. But they didn't stop here. They continued to work with arginine mutants, and they identified 3 different classes of arginine mutants by supplementing these mutants with different classes of basically different precursors of arginine. What a precursor is in a metabolic pathway, you have the final product. Here, so you have the final product, which is arginine, but you don't go from A, which is your original nutrient, to B, which is the final product immediately. There's a series of reactions and series of intermediate products that the organism needs to create to get to the final amino acid, which in this case is arginine. From the precursor to each of these steps is an enzyme, or an enzyme catalyzes the reaction. So what they mean by different classes of arginine mutants is that in different classes of arginine mutants, different enzymes in this pathway are not working. So you could have an arginine mutant that has this enzyme not working. You could have an arginine mutant that has this enzyme or this enzyme not working. But the final result is that they're all arginine mutants. But you can say that they are different classes because they have a different enzyme that's broken. So a different gene is broken. They're not the same. So this experiment, the goal was to identify at the step at which the arginine metabolic pathway was blocked by the mutation. So how did they determine this? They took different classes of mutants and they supplemented them with basically different substances on the pathway. So we'll look here. Minimal media would be the negative control, so it has none of the precursors. Arginine is the final product. Citrulline, if you see here, is second last. Ornithine is, you know, third last. Sounds weird to say. And then there's just the precursor. If it can grow with just the precursor, then it's not an arginine pathway mutant because that means that if it can grow off just the precursor, then that means this is working, and this is working, which is why they don't supplement the precursor here. So let's see what this means. Mutant number 1. It can grow with, or sorry, I take that back. Let me correct myself. If it can grow with ornithine, citrulline, and arginine, then that means that it is like there's an enzyme that creates the precursor that's broken. And I'll explain it in just a second. Mutant number one. It can't grow on minimal media, but it can grow on arginine, citrulline, and ornithine. So what does that mean? If it can grow on arginine and citrulline and ornithine, but it can't grow on minimal media, which let's say is like back here, then what it means is that there is something here, or it's like this enzyme is broken. Because let's say that minimal media is a precursor. Then this enzyme, oops, then this enzyme, enzyme number one, is what's broken. Because it can work with ornithine. It has enzyme 2. It can create citrulline. It has enzyme 3. It can create arginine to continue, if that makes sense. Mutant number 2. It can't grow on minimal media. It can grow on arginine. It can grow on citrulline, but it can't grow with minimal media and ornithine. So what does that mean? It can grow starting here. It can grow starting here, but it can't grow starting here. So when it has only ornithine, pathway 2 is not working. Mutant number 3, it can only grow with arginine. So even if you give it citrulline, ornithine, or the precursor, it won't work. So this enzyme is not working. This is basically what sums up this experiment. Yeah, this little chart. I know it's a lot to take in right now. For mutant 2, does the enzyme not work in precursor as well? Yeah. So we can say that, let's say that the precursor is the minimal media. If you look over here, the minimal media is the negative control. So mutant 2, mutant 3, both don't work with just the precursor. Actually, no, that's wrong. I take that back. The precursor is not the minimal media. The precursor is like not mentioned here because it's basically the first step on the pathway. Yeah, Shams, sorry for confusing you. Mutant number 1 works on the precursor because it works on arginine, citrulline, and ornithine. Mutant number 2 doesn't. So mutant number 2 is blocked already at ornithine. So the precursor, which only goes into ornithine, it can't go past it. So if we said that mutant number 2 is not working at enzyme number 2, it doesn't matter if it has enzyme number 1 because it can't get past the point anyway. Does that make sense? Yeah. Yeah, sorry guys. The precursor is not the minimal media. I take that back. Alright, let's continue. I know this is a lot to take in. If you guys have any questions, do ask. If you have questions like tomorrow, like still ask. Let's say you had a test. You're going to be given like sample data like this, and you're gonna have to be able to like basically categorize mutant number 1, mutant number 2, mutant number 3, and be able to tell where on the pathway the enzyme is not working, just based off a chart like this. So, right, yes, so this is basically just summarizing everything. Cool. So what did they conclude? They showed how genes are related to proteins. Because each mutant was defective in a single gene, mutations in genes affected production of enzymes, and this implied that metabolic diseases can be inherited. So their experiment also supports the one gene, one enzyme hypothesis. But the one gene, one enzyme hypothesis is actually inaccurate. So could you guys name some other possible statements? So if we had, let's say, one gene, one protein. How is one gene, one protein different from one gene, one enzyme? So let's say one gene, one enzyme. Oh, one gene, one enzyme. How is that different from one gene, one protein? And I want you guys to think about examples like, uh, yes, that's a good point. What about one protein always being one enzyme? Is that true? Exactly. Okay. What about four proteins per enzyme? Mm-hmm. What if one gene could code for more than one protein? Two proteins. Oops. Which we might see today. Hint, hint. All right, let's move on. So, the point of this slide is to basically say that one gene, one enzyme sounds pretty good on the surface, but there's a lot of other things hiding under here that you might not think about. Is one protein always one polypeptide? So also that to think about. We'll think about that in a second. Yeah, so here, not all enzymes are proteins. One protein may have more than one subunit. Some genes do not produce polypeptides. And one gene may produce multiple polypeptides. And we'll learn about splicing today. There's some videos. Right, so before we go further, we need to talk about something called the central dogma. And the central dogma is basically a diagram that shows how is DNA, mRNA, and protein related. So here is a very, very simple example of what could be a central dogma. DNA is converted to mRNA in the nucleus through a process called transcription. mRNA is converted to proteins either in the cytoplasm or rough endoplasmic reticulum through translation. But this is only a portion of the actual central dogma. Here is another example. It's basically the same thing we saw above. Here is Watson's central dogma. This isn't actually the original one, and there's some errors in this representation. Can you guys spot any of them? Any ideas how we could change this? Can RNA make more RNA? Ooh, ooh. Let's scroll down. Good question. There's lots of different central dogmas here. Only one of them is correct. Let's say that this is one, this is two, this is three, and this is four. Any ideas? Anybody think one is correct? Can proteins drive more proteins? Proteins make DNA? DNA directly to protein? Easily? All the time? No. One is not correct. Let's look over here. Yes. Alright. I'll reveal the answer in just a second. Let's look at number 4. Dotted means that it can happen, but it's more rare. Solid basically means that It can happen regularly, normally. Yeah, that's basically what it means. So dotted is like, it can happen, it's possible, but it's more rare, if I to say it. Yeah, I'll just go ahead and say it. So yeah, RNA to DNA, the dotted line basically represents that it's rare because this only happens with a reverse transcriptase. DNA to protein, even rare. I don't think they have an example of a live organism doing this. I think they've only created it in laboratory situations, to the best of my knowledge. They had to like carefully control the environment to get this working, but it can happen. So yeah, example 2 is actually the correct central dogma. This third one is missing RNA guided RNA replication and reverse transcription and DNA guided protein synthesis. Same thing here. Or sorry, the dotted lines. Same thing here. This one's also missing reverse transcription. All right, let's move on. So this is actually called Crick's central dogma. So if you guys notice these names, the previous one we saw was Watson. This one is Crick's. Yep. Right. That's a little video. So this is just to give you guys a little mental image or place yourselves as to where eukaryotic protein synthesis actually happens. So we know that DNA replication happens in the nucleus, but transcription also occurs in the nucleus, and eukaryotes have this extra step called RNA processing that occurs in the nucleus. And afterwards, translation can occur outside of the nucleus. Prokaryotes have it a little bit simpler. They don't have a nucleus, if I remember correctly. They just have a nucleoid region. Transcription and translation occur in the cytoplasm at the same time. There is no such thing as RNA processing for prokaryotes. So I'll go back, take a look over here, and take a look over here. Cool. So here, I think this entire thing is a nucleus, if I'm looking at it correctly, and everything outside is the cytoplasm. But here it's just this is the cell and this is the cytoplasm. Right? Cool. Let's go over some definitions. What is the gene or a gene? The gene is the stretch of DNA that is transcribed into RNA. It's also called a transcription unit. Notice that transcription begins here. When you refer to specific positions, the beginning of transcription is +1. I don't think there is a 0 position, but I may be wrong. Right. Let's look at this overview. Just like DNA replication, there are 3 stages to transcription. Yes, one indexed. Uh, there's initiation, elongation, and termination. Is a little overview. Alright, so for pretty much most of the things that we're going to cover from now on, eukaryotes and prokaryotes have both have a promoter, but the promoters for eukaryotes are not the same as the promoters for prokaryotes. So by the end of this unit, you will have to make a chart comparing and contrasting prokaryotes and eukaryotes. So let's continue. The promoter, the general definition, is where RNA polymerase first binds. This is not the same RNAP involved in DNA replication, I believe. Oh, or prokaryotes only have one RNAP, so maybe it is the same one. Prokaryotes have one RNAP, eukaryotes have three. The promoter is located upstream of the gene. So what does that mean? Let's go back here. Upstream means before the start of transcription. Downstream means after. Let's go down. Yeah, one indexed but with negative numbers. Indeed. So the promoter is located upstream of the gene, usually composed of an AT-rich region. Can you guys think of why A and T? AT, AT, CG. Why not CG? Any guesses? It's quite simple. Yes, exactly. AT has two hydrogen bonds, so it is easier to break. That is the exact reason. Prokaryotes have something called a Pribno box. Eukaryotes have the Tata box. Very easy to remember the sequence for eukaryotes because Tata, and then for the Pribno box it's Tatat. So that's how I remembered it. The numbers are there. I don't remember having to remember the exact numbers, but it's always good if you can add it to your chart later. So the promoter contains the transcription start point. This is important. And the start point is where RNAP starts transcribing. Initiation processes are different between prokaryotes and eukaryotes. Prokaryotes, the RNAP recognizes and binds to the promoter. It unwinds the DNA and then begins transcription at the start site. But for eukaryotes, there's a complicated process where a transcription pre-initiation complex has to form by the following components binding in order. First, transcription factors, which are proteins that help initiate and regulate transcription, need to bind. So it's what's going on over here. And then an inactive RNAP2 has to bind onto the promoter. So all of these little circles that are not the big one, or that are not this like big white thing, are transcription factors, and this big white thing is RNAP2. So as you can see, many more steps involved. For eukaryotes specifically, the RNAP2 is activated by phosphorylation on or phosphorylation of the C-terminal domain. I don't know where the C-terminal domain is, so this is just something I know that happens, but I'm not too sure where and how it happens. So transcription begins once the CTD on RNAP2 is phosphorylated. So over here. Wait, no, let's go. Other factors. Yes, phosphorylation is going on over here. And then it chugs along. So let's define some strands here. The coding strand is also called the sense strand. The non-coding strand, also called the antisense strand, is also called the template. And transcripts are newly synthesized RNA. So let's take a look at this in the little diagram here. Right, this section. Is this the transcript, the template strand, the coding strand, the sense strand, or the antisense strand? Can anybody tell me? Maybe I'll go back a little bit. You guys take a look. The coding strand, oh, good. So it's the template and the antisense strand and the non-coding strand. Which is weird. It was always weird to me that it's called a non-coding, but I guess since it's not directly bonding, it's called non-coding, but like this is actually exactly what the oh sorry, I'm not gonna say what it is. Right, good job. What is this bottom blue strand? Good. Coding strand, also called the sense strand. And this pink. Yep. Can you guys name 5 prime and 3 prime? So over here, let's say over here, would this be the 3 prime end of the transcript or the 5 prime end of the transcript? Yeah, so it is a 3 prime end, and the way you know is because the diagram is telling you that transcription is going this way. We know that RNAP needs, or sorry, RNA polymerization will continue by adding on to the 3 prime end. Polymerization happens by adding to the 3 prime end. Where can we see this? What gives us a good look? I think, yeah. So, what do you mean? Oh, I was talking about this this pink thing here. So it's adding to like 3 prime. It's polymerizing. So this is the 3 prime end. So on the DNA. So let's ignore the pink for now. On the DNA, let's just like make boundaries. This is a little bit hard to describe, but let's say over here, and if we said this was an end and this was an end, is this 3 prime or 5 prime on the right side here? Yep, good. So which means this is, and anti-parallel, we can know these ones. So this one should be 5, and this one should be 3. So whenever you're given a question, you have to name the diagram like so. I would always recommend, first of all, you look at the direction of transcription. So you like place yourself in orientation. Once you know the direction of, so is downstream always 3 prime to 5 prime on the bottom strand? DNA? Bottom strand of DNA or bottom strand of, or just strand of RNA? Yeah, pretty much. Because a better way to say it is transcription always happens downstream and it always happens toward 3 prime end of RNA because you always have to add to the 3 prime end of RNA. So just based off that knowledge, you know the direction on this strand and then you know the direction of this strand. So it's like deduction. Any questions? Okay, cool. So now we are at elongation. RNAP unwinds DNA, exposing 10 to 20 bases at a time. It uses the template, which is the bottom strand over here, to, wait, did I say that right? I think the template is the top or the bottom? Give me a second. Complementary. What have they said here? Template strand is the bottom. That's what it says. So I guess I took the wrong answer. The template is actually this one. I took the wrong answer previously, guys. So yeah, okay, so that's why I was actually thinking of the right answer, wasn't I? Okay, sorry guys. This is the template and this is non-coding because if you look at the actual bases, this is a C and this is a G, right? So if we call this a non-coding strand and this is a coding strand, it makes a little bit of sense because if you look here, if you look at corresponding bases, this is a C, this is a C on the RNA, and this is also a C on the DNA. Obviously thymine becomes uracil, but A is A, C matches C, and then a G would match G eventually. So this is why we call the top the coding strand and the bottom the non-coding strand. But it's also the template because it's what the RNA is built off of. Yeah, this is all kind of coming back to me as I'm teaching you guys too. Sorry about the confusion. So I'm just going to say it one more time. I took the wrong answer previously. The template strand is the strand directly binded to the RNA. It's also called the non-coding strand, also called the antisense strand. The top strand, which is not interacting with RNA, is called the coding strand or the sense strand because the bases correspond. So C is C here, C is C here, A is A, T is just U just because of the differences, but G is also G. So, any questions? All right, cool. So, as we continue along, so as RNAP continues along the DNA, the RNA transcript separates from the template as RNAP continues the elongation process. And the DNA helix reforms behind RNAP. So if you can see here, it's reforming over here, unwinding over here, and the transcript is separating. Let's say that RNAP gets to the end of the gene. What happens then? How is transcription terminated? So prokaryotes and eukaryotes have a separate process. Let's look at the prokaryotes first. For prokaryotes, there are two things that happen. A GC-rich region, NTP. Where did you see NTP? Oh, nucleotide triphosphate. Yep. So it's like a nucleotide, but it has three phosphates attached to it. I remember earlier when yesterday's lesson where we discussed that the three phosphates, two of them are broken off, used as energy, and then one of them participates in the polymerization to continue the backbone. That's why it's a triphosphate. Does that make sense? Yeah, cool. So, transcription termination in prokaryotes. Two things have to happen. First of all, a GC-rich region comes along. And earlier, transcription began at an AT-rich region on the DNA. Here, it's GC-rich. So what do you guys think is happening? They are, but what actually happens is GCs, yes, they have, it's more like they have stronger interactions because of the three hydrogen bonds that can form, but this GC rich region is actually referring to the transcript in terms of what's going to happen. So take a look at this diagram. Here's a GC rich region. Here's another GC rich region, which means that you have two GC rich regions on the transcript. So what happens is, as it detaches from DNA, separates the interactions between these two GC rich regions is going to create something called a hairpin loop. So this big thing is called a hairpin loop. And this also causes RNAP to stall. Does that make sense? And right after, or after the GC rich region, there's something called a deoxyadenine uracil rich region. Deoxyadenine is called, we're using deoxyadenine because GC is referring kind of to the transcript. So if you look here, it's basically saying because on one end, on the transcript, it's going to be uracil, and the other end, you're going to have a deoxyadenine. Would there be more lines of GC? If there are more GC rich regions, would there be more lines of GC on the hairpin loops? So would there be multiple hairpin loops, or are you saying that instead of something like this, you're also going to have another section like this? Is that your question? To be honest with you, I don't know. I haven't seen that, but that's a good question. Maybe ask Miss Law. Right, so, right, cool. The deoxyadenine uracil rich region is downstream on the RNA. This region has particularly weak interactions, two hydrogen bonds. So this is where RNAP stops transcribing and RNA is actually released from the RNAP here. So here's a little diagram. This shows pretty well. Okay, so eukaryotes. First of all, there's a dephosphorylation of the CTD on RNAP2. This reduces the rate of elongation and it also increases pausing. And afterwards, eukaryotes also have sequences. So there's something called a terminator sequence. If you can look over here, the terminator sequence is after the stop codon in something called the trailer. The terminator sequence is on the nascent RNA, so it's on the transcript. AAUAAA, oh, CTD, it's nascent is newly created. It's like basically nascent RNA is equals transcript. CTD, let's go all the way back up here. When we started transcription, boom, 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 here. The C terminal domain, it's like this little tail thingy on RNAP2. You see here? This is the, not ignoring the phosphates, this little like region here, this is what the CTD is. Nascent means, yeah, newly created. So AAUAAA means this is on your RNA. This sequence stalls and pauses RNAP2, but RNAP2 can still continue for a few bases to thousands of bases past this terminator sequence. What it actually stops is this GU-rich sequence. And once again, this is referring to the RNA itself. GU will be on the RNA itself. This is 40 to 60 nucleotides downstream of the termination sequence. And this sequence signals the cleavage of RNA. So this is a little bit different than prokaryotes because in prokaryotes the RNA just like falls off, but here an endonuclease will come and cleave your transcript. So the RNA cleavage site is between the two sequences mentioned earlier, and it's about 10 to 35 nucleotides past the termination signal, which is this, and this is what releases RNA from RNAP2. Very different from prokaryotes. So let's do a quick summary. Lots of components. Let's take a look. I can't really tell if this is a prokaryote or a eukaryote just off of this information, but can we identify where the prokaryote and eukaryotes differ? Let's see, binding of RNA polymerase and local DNA unwinding. Eukaryotes and prokaryotes do not bind RNA polymerase the same way. Remember earlier that eukaryotes require transcription factors to finally bind RNA polymerase. For prokaryotes, they don't need transcription factors. RNA polymerase binds at the promoter on the Pribnow box, or at the Pribnow box. Let's check over here. Initiation of RNA synthesis. I think that was also different because eukaryotes require, or eukaryotes have the RNAP phosphorylated. Elongation seemed pretty much the same, right? Elongation, there wasn't really a difference. Let's continue. Termination. Termination was very different between eukaryotes and prokaryotes. We just covered that. Eukaryotes have a terminator sequence and a GU-rich sequence, and then the RNA is cleaved in between them. Prokaryotes have this GC-rich region that creates hairpin loops, and then a DAU-rich region where the RNA is released. Alright, so there is a difference between prokaryotes and eukaryotes after or during this transcription too. So let's take a look at this example. Or no, this is not it. I think this could be the same for prokaryotes and eukaryotes. Sorry, I got ahead of myself. Alright, so let's look at this. Let's say that you have more than one RNA polymerase on the same DNA strand working at the same time. What's basically going on here is they're in a line and they're transcribing in a line because just because you have one RNAP working on the DNA doesn't mean that you can't have other RNAPs working before or after it. So many RNAPs can transcribe simultaneously on the same gene. So as long as the promoter is free of RNAP, then the next RNAP can bind and begin transcription. So there's no need to wait for an RNAP to complete transcribing the whole gene before the next one begins. And this is important if you need to create lots and lots and lots of copies of this transcript. Let's take a look. The blue line is, let's take a look here. dsDNA. Red line is ssRNA. Green. I don't know. I'm not too sure what this DS and SS means, but the blue line is DNA, the red line is RNA, and the green dots are RNAP. So if you take a look here, let's say that this is like one gene from like here to here. Look at all the different RNAPs that are on the gene at the same time. Oh, that's a good point. Yes, that's probably what it means. Thank you. Double-stranded and single-stranded. Thank you. So let's think about this a little bit. Can you guys tell from this picture what direction each RNAP is traveling in the diagram? Is it traveling down or up? Any ideas? Down. Okay. Okay. Good. So, you guys are right. The ones near the bottom of the page, the RNA is longer or even completed, which suggests that it's further along the gene, so they're moving down. Right, cool. So, the changing of RNA. When you have just finished transcription, we call that RNA the primary transcript. I think RNA processing is unique to eukaryotes. So I think a lot of what's being said, yeah. So what's being said here is only going to apply to eukaryotes. In eukaryotes, when RNA is done transcribing, at first it's called the primary transcript, or also called pre-mRNA or raw mRNA. mRNA is called messenger RNA, and this is the final transcript after all processing has occurred, and mRNA is what gets exported out of the nucleus. So for a little bit of a description here, this is your primary transcript from 1 all the way to position 146. Have you guys learned what exons and introns are? Probably not yet, right? This is just a diagram. Okay, cool. 3 things happen during RNA processing: capping, polyadenylation, and splicing. All 3 things are very important. Let's look at capping first. As you can tell by the name, a 5' cap is added to the 5' end of your primary transcript, or I'm going to call it the pre-mRNA from now. It's added by a capping enzyme complex. The 5' cap itself is a modified guanine, and it protects mRNA from degradation, and it also signals ribosome attachment. And we'll learn what ribosomes are, I think not today, but maybe the next lesson. Important for translation. So take a look. This is the structure of the 5' cap. This is the 5' end of DNA. Oh, sorry, mRNA. And this is the 5' cap. Cool. Next step, polyadenylation. So polyadenylation is when a poly A tail is added to the 3' end of your pre-mRNA. The poly A tail is 50 to 250 adenines. It's added by poly A polymerase. What does it do? It protects mRNA from degradation, and it facilitates the export of mRNA from the nucleus. Let's take a look here. Can capping happen during transcription? Before the RNAP is done? Anybody have any guesses? Or based on this diagram here? So let's say that the 5' cap, so capping only refers to the 5' cap because the poly A tail, adding that is called polyadenylation. So when we say capping, we're referring to the 5' cap. So exactly, so anytime after the RNAP has begun, like the 5' end is free, right? Nothing's going on here, so the cap is added. Can polyadenylation occur before the RNAP is done? No, it can't. So take a look at this diagram. So after cleavage, then poly A polymerase comes in and it adds your poly A tail. This is the poly A tail on the 3' end, and this is your 5' cap on the 5' end. Cool. Now we will get into something called splicing. Splicing is the removal of introns. Introns are intervening or non-coding sequences, and they are interspersed, so spread out, between exons. Exons are the coding sequences, and they are expressed. Expressed means that they are translated. Or, I guess that's a good way to say it. Or, actually, yeah, there are two examples of exons that are not translated. So let's look at two exceptions. Exons are expressed rule, right? But there's a leader and something called a trailer. And these are exons that are not translated. So let's look at this position 1, and position 146. If you look over here, between the 5' cap and the start point, and position 1, there's this little section here called the 5' UTR. UTR means untranslated region. 3' here, boom, 3' untranslated region. So these are called the leader and the trailer, respectively. How would you identify an exon that is untranslated? These are the only two examples. The only exons that are untranslated ever is the leader and the trailer. So let's take a look at, let's scroll way up. Yeah, actually maybe even further up. Oh, I think I went too far up. Sorry, this might be a little bit dizzying. Alright, so this is position 1. Remember that the promoter is like somewhere here. And then there's like a start point. Oh, actually maybe it's better to look it over here. Cool. You see that there's like a promoter here and there's a start point here. But then there's also like this section or this section. So what's going on here? Let's take a look. Okay, I guess we'll take it back here. All right. Sorry, moving along a lot. None of these diagrams are too good to explain this, but essentially, once you have this RNA, you will learn in translation that all your codons, or this is your coding segment, And this section is what's going to be turned into your polypeptide chains. So then once you have that information, it'll be much easier to identify your leader and your trailer. But for now, you just have to know that the leader and the trailer are not spliced, even though they're not translated in the, like, even though they're not expressed in the end, if that makes sense. Does that answer your question? And David will answer your question of why introns existing, even though they're not translated, in just a second, because they're very important. So what is splicing? Inside the nucleus, there are proteins and small nuclear RNA called SNRPs, plus other proteins. So other proteins, SNRPs, they come together to create something called a spliceosome. What spliceosomes do is they excise introns from your pre-mRNA. How do they do this? There is something called a splice site. It's a short nucleotide sequence at the ends of introns. This is where your small nuclear ribonucleoproteins bind, and this is where excision occurs. So you see like the pink little line here, these are RNA enzymes. Does that make sense? So these are like RNA strands that are acting as enzymes to slice your pre-mRNA. The way it kind of works is imagine that the spliceosome grabs these splice sites, pulls them closer together, and then pinches it off, pretty much. I don't know if there's a diagram anywhere, um, but maybe there is. No, I don't think there's a good video or diagram on the page here, but you're gonna have to take my word for it. So these ribozymes, or the ribozymes, oh, excise means basically like cut off, removed. It's like, have you guys ever like burnt part of your food and then, or burnt part of your toast or something like that, and then you don't want to eat the burnt part, so you like cut it off? It's kind of like that, excised. Or let's say um, you had a string and you want to cut off a section of the string. So what you do is you cut off both sections that, or, so, I mean, I can just draw it. So let's say you have like a string here. You cut this section and this section, and then you glue these two parts together. That's basically what's going on. This is the intron, and it's being removed. The splice sites would be over here. Anybody confused as to what spliceosomes are doing? Just the most important things to take away here are that SNRPs are the functional units of the spliceosome. The small nuclear RNA are ribozymes, and they are doing the cutting. Cool. So they excise introns and rejoin exons. There is evidence for co-transcriptional splicing. So as transcription is happening, splicing is also happening at the same time. And here's a little picture. So if we thought about the diagram that we just saw with all the RNAPs moving along the DNA, if you can see here, there are little pinching. There's like little pinching that's happening. You can see that. You can circle them out. So the sections that are being pinched off, we can assume to be introns. And you can see a little bit of a darker, larger circle. Maybe that's the spliceosome. Yeah. All right, so why do introns exist if we're just going to cut them out? Why go through all the work of transcribing them in the first place? And the best way to describe why introns exist is alternative splicing. And basically, alternative splicing allows one gene to code for more than one protein. So how does that happen? Using alternative splicing, a single gene can encode more than one kind of polypeptide depending on which segments are treated as exons. So this has a regulatory role. It controls gene expression, quite literally, and it occurs at the RNA level. So let's take an example. Let's take this section of DNA and these 5 exons. Let's say your body, or yeah, your body treats, in the first example, all 5 exons as exons, and they're all kept. In the second example, exon number 3 has been considered an intron and spliced out. In example number 2. Example number 3, exon number 4 has been considered an intron and spliced out. The resulting proteins from the different polypeptide chains are different. They have different shapes, which means that they would have different functions even. So from one gene, you have created 3 different proteins, or 3 different polypeptide chains. That's a better way to put it. Another function of introns is to help exon shuffling. Introns increase the probability of beneficial crossing over or the recombination between 2 alleles. So this occurs at the DNA level during meiosis. So do you guys remember the crossing over phase of meiosis? Where DNA comes together, they like attach, and then they basically exchange sections. Let's take a look at this. So let's take a look at that over here. Gene 1 and gene 2. Let's say that they attach like at this point. So let me draw a little line here. And they swap this section during meiosis. What happens? Gene 1 that originally had exon 2 has moved exon 2 to gene 2. But could this have happened if this entire section was an exon? Or rather, the existence of intron 2 and intron 3 and intron 2 on gene 2 allowed like this point of crossing over to switch to basically transfer gene or exon 2 from gene 1 to gene 2. Does that make sense? So this allows each gene to have different exons or change their protein domains without affecting other coding sequences. So this exchange of exon 2 from gene 1 to gene 2 did not disturb any other exons on either genes because the introns were there. Does that make sense, guys? Um, yes. Yeah, it is. So if you look at the proteins in themselves, like protein 1 and protein 2 before and after, yes, protein 1 is missing that section of it. Look at this. Domain 2 is missing. Natural selection may eliminate this gene from the population if it is no longer functional. A protein with domain 2 from gene 1 inserted into gene 2. If this protein provides a new beneficial trait, natural selection may increase its prevalence in a population. So remember, like, diversity and a larger gene pool is always welcome. And this is just one way that it increases that. It just adds more randomness, I guess. Which is, yeah. So let's take a look. Prokaryotic versus eukaryotic protein synthesis. We've covered up to DNA replication and transcription so far, but we're going to have to cover translation in the next lesson. But this gives you a quick overview of what you might expect. So, in prokaryotic cells, we've learned that transcription happens basically in the same cytoplasm as translation. just through this diagram. So it's much simpler. No RNA processing happens, and this diagram is basically there to show you. DNA to mRNA to ribosomes, and then a polypeptide comes out. But in eukaryotes, it's much more complicated, where once a pre-mRNA is created, it needs to go through processing, get turned into an mRNA, which can then be shuttled out of the nucleus to a ribosome to create your polypeptide chain. Lots more work, but for a good reason. And that's the end of the lesson. Here's some homework questions. Do you guys have any questions? Anything super confusing? Oh, cool. All right. I'm gonna give you guys a little video in the chat. This is from Miss Law on our website, so you can take a little look at it. It's another one of those like 3D model thingies of proteins working. I thought it was pretty cool. It's 4 minutes long. You can take a look at it. So yeah, if you guys run into any questions, I recommend asking because this is gonna keep coming up. All of the concepts here, they're very important. And by the end of this, I recommend that you guys create a chart. I think in the next PowerPoint there is a chart example. If not, I will try and create a chart for tomorrow so that you guys can like kind of copy the foundation of and then fill in yourselves. And yeah, if there are no more questions, that's it for me. So, any more questions, guys? Alright, cool.